chart, and then so today I'm going to talk about the evolutionary transition and my most recent computational simulation demonstrating the ETI. So this is part of the project, the scaling up purpose in evolution, funded by John Templeton, and the PI is Richard Watson. So as we already knew that ETIs refer to the key events in the evolutionary history where organisms evolve new levels of organizational complexity. This process often involves the integration of previously independent biological units into a new and cohesive entity. And a classic example is a transition from the single-cell life to multicellular organisms, and also the evolution of eusociality or eusocial superorganisms. And the program was set like 30 years ago in the classic book Major Transition in Evolution. The evolutionary puzzle here is that the classic evolution by natural selection theory, focusing on the combination between self-interest units. So it's harder to imagine how natural selection can explain the emergence of and evolution of new higher level units from interactions among lower level entities. And here we are facing circular problem or chicken and egg reasoning, because most existing theories often presuppose the existence of higher level units, which only emerge after the transition. And so the process of ETI can be divided into two phases: the pre-transition phase or the group formation phase. And some key questions in this phase that how do individuals, entities initially come together to form cooperative or coordinated groups, and what mechanisms drive this aggregation? Is this purely random, or is there any underlying biological process? And the second phase is what we call post-transition phase, or the group transformation. So since the aggregation of the group is formed, there is a transformation process of that group into a more structured, integrated organization as a new level of individuality. And some key questions here is, what processes guide the transformation of this transformation? And yeah. So our plan is that today we'll explore the mechanisms behind ETIs, talking about some limitations of current theories, and I will introduce my approach based on the combination of self-organization and emergent selection processes, and which will be demonstrated in an artificial life simulation. So yeah, several theories of current viewpoints of ETI. The first and famous one is kin selection. This says that natural selection favors the reproductive success of an organism's relatives, and some famous proponents like W.D. Hamilton. So it has the strength of providing a robust explanation for the evolution of altruistic behaviors in a closely related group, but it's not adequate enough to explain the cooperative behavior among unrelated individuals. And the second one is more controversial one, is what we call group selection. And the basic idea is that natural selection will operate at the level of the group rather than the individual. Some proponents will be like Dave Wilson, and it has the strength of explaining how traits beneficial to the group can evolve, even if they are costly to individuals. But this theory is very debatable, and a lot of debates in the history of biology, like between Richard Dawkins and Dave Sloan Wilson, or even E.O. Wilson about this. Yeah. So this debate encapsulates the fundamental question in EvoBio. So does natural selection act primarily at the level of the genes? It's more reductionist view, individuals or groups. Another viewpoint is the fitness decoupling proposed by Richard Mitchell. And this is a process where the fitness of individual components is integrated into the success of the entire group. and it has the strength of allowing for a higher level of specialization and division of labor. Yeah, but it's, of course, it has some weakness too, and the most obvious one is that it does not explain the initial steps toward forming these in the interdependent relationships between entities. And all these theories and viewpoints are post-transition focus, and it highlights the gaps in the research of ETI, because it does not explain how the initial group is formed, what is the mechanism behind it, how is the drive of it. Another viewpoint is called ecological scaffoldings. This means that environmental structures and interactions that facilitate emergence of new biological individuality by providing some scaffoldings in the environment, right? So, yeah, and it's crucial in the early stage of the transition. Some mechanisms of this is like physical structures, like it can provide a boundary to protect new groups, and the concept of niche construction is here. Or also, this means that organisms modify their own and each other's environment or niches, leading to new selective pressures. For example, like it can change the environment in a way that reduce the selective pressure that foster more coordinated or cooperative behavior in the first place. Yeah, some classic examples of ecological scaffolding in bacterial biofilms or eukaryotic cell evolution. Yeah, of course, it has some limitations too, because it's often presuppose extrinsic conditions, and the ecological scaffolding more effectively explains how certain environmental structures can support the maintenance of higher level individuals once they are established, and less clarity on how these structures emerge. And it has both theoretical gap and experimental gap in this viewpoint. And a more recent viewpoint proposed, supported by my PI in Southampton and also with Mike in the paper, they call it connectionist approach to ETIs. So the basic idea here is that they saw some analogy between evolution and learning as an optimization process, or they call it the ball rolling downhill. And so this means that because evolution is analogous to learning in neural networks, so they saw the similarity between the ETI and the process of hierarchical concept formation in deep learning. And the idea is that self-organization can drive adaptation autonomously without resorting to natural selection as a primary drive in evolution. Yeah, so in the most recent paper, like they call it natural induction, spontaneous adaptive organism without natural selection. It's a really cool idea. But of course it has limitations too, based on my perspective, because the biggest difference here is that the structure of most deep learning of neural networks is predefined by the experimenter rather than a spontaneous emergence in ETI. And there is no inheritance mechanism at the component level, and also the reproduction process as a component level to say that there is a transition in individuality in Darwinian sense. So it also means a transition in level of inheritance and reproduction, and it lacks some empirical support too. And so it goes back to the debate between self-organization and natural selection as the main drive of biological adaptation, like the debate between Stuart Kaufman and Maynard Smith years and years ago. But I, but here I think that self-organization and natural selection can be complementary rather than mutually exclusive. For example, the easiest way to think about it is self-organization can initiate the formation of complex structures, which natural selection then can act on it by refining and stabilizing it over time. Or another way, the ability to self-organize is an evolvable adaptation. This means that we are talking about the evolution of self-organization. Yes, it's this evolutionary process can promote the emergence of collective self-organization as an adaptive response to environmental challenges. So I propose that by combining natural selection and self-organization, we can study the spontaneous emergence of pre-transition group formation and also the spontaneous emergence of group selection, post-transition selection. Yeah, so I use an artificial life approach to ATI, as we may know that a life is the study of life as it could be rather than life as we know it. So we study the basic underlying fundamental processes and mechanisms of life, regardless of the material it's based on. So it allows us to rewind the tape of life in the computation or by artificial chemistry. Yeah, and I think a lab is a unique platform in order to test and observe how complex interactions and evolutionary pressures can generate higher levels of biological organizations as in ATI. Right. Oh, and actually, there have been many models of self-organizations in a life, like um, cellular automata-like model, or like Conway's Game of Life, or Langton's Loop, or even the most recent one, linear. So in all those models, we have the formation of patterns and the replicating process of patterns over time. But they are not adequate to say that it's an evolutionary transition in individuality. The same reason as in Richard's conditionalist approach, because there is no inheritance at the component level and no selection at the component level also. So by this, I mean that in order to model an ETI in an artificial system, we need inheritance, selection, and reproduction at the component level. And by some way, these combination can give rise to an emergence of the reproduction or replication at the group level, as if the new level of individuality emerged. And then, so I developed a life model. I call it Vita Nova. It means new life in Latin. And in this simulation, there is no explicit fitness function. So we use the fitness as an emergent property, and selection here is also an emergent property from the interactions of lower level units. So it's closer to biological natural selection rather than using the fitness function in normal evolutionary computation. And here's a design methodology of Vita Nova. So we have 2D square and some agents I call boys. Yeah, so the green boy is the worker boy and the blue boy is the soldier boy. And the red one is a predator, randomly distributed in the environment. And each boy is controlled by its own neural network, which will modulate the core steering behaviors of boys. Like we know that it's separation, alignment, and cohesion. So it's different from the classic boy simulation of Reynolds when he used the prefixed parameters to for these behaviors. The inputs to the neural network will include the relative positions and velocities of nearby boys and predators, and also the current energy level. The output, as we know, is a tendency for separation, alignment, and cohesion, and also predator avoidance and the role tendency. The role tendency here is the likelihood of being a worker or soldier in the next step. This means that in my simulation that we have plastic division of labor governed by neural network, exactly the evolving neural network. And the behavior of the predator is that to detect and move towards nearby boys or reverse the direction if facing three or more soldier boys. This is the setup of the simulation. Here's the basic architecture of the neural network. It's a simple feed-forward neural network with one hidden layer and equations for boys' behavior. And here's a pseudocode for reproduction and mutation. So there is no explicit selection. So the reproduction here is so in this current simulations, there's no autonomous, there's no neural network output to control the reproduction. So when the boy reaches a certain amount of energy, it will reproduce and reduce some energy. And the reproduction is a replication with some mutation in the weights of the neural network. Yeah, so the neural network's weights are the genotype of boys, which are evolving. And the concept of fitness here is just an emergent concept. And here's the video demonstration of boys of my Viton Nova. So initially we saw that they are trying to, as I put some statistics on this, the number of flocks and average flock size. Some are trying to form flocks, but not good enough. Yeah, so most flocks are destroyed uh, until like one flock in the corner survives. Yeah, so it moves toward the corner. And here, I think it starts growing, or we can call it kind of development until an adult stage. Um, yeah, so there is a ring structure here. It means like there is something inside and on the circle. Yeah, it starts growing. The average flock size is increasing. Yeah, some predators around to put pressure on it. There are some blues here to block the predators from attacking the flock. Yeah, it's growing, it's developing. And the critical moment is around steps 3,200, I think. Yes, yeah, suddenly it starts spreading into two entities, and the neuron structure emerges here. And also it reproduces again. A neuron structure moves toward the screen. Yeah, they move in different direction. And oh yeah, sorry. Yeah, we can we can call that the first flop in the on the screen is flop 0, and the first ring structure is flop 1. And the flop 1, after moving, it starts reproducing again into two ring structures. Right. And also you see it the second child, yeah, it starts reproducing again in the bottom of the screen. Okay, so yeah, so the result overview is that we have observed emergence of cohesive groups, a transition from solitary boys to organized flocks, and the formation of ring structures, exhibiting both stability and capacity for division, a potential mechanism for group level reproduction. And also, we have observed the adaptation and survival at the group level. And there is some a spontaneous emergence of group selection because some groups are formed, but they are not good enough to survive. And the only one survives, then reproduces itself. Some snapshots of the screen, some critical moment. We observed it. Yeah, so it's just statistics. We observed that there is number of flock count over time until the step of like 2,000 times step. Only one flock survived for more than 1,000 times steps, and the flock size starts increasing until the critical point of the transit of the reproduction. It reduces the size because there are now two flocks. Yes, and again, it supports a hypothesis and observation. And here we observe the another, we call it, I put this figure for observing the role distribution and collective adaptation. So here we see that the soldier and worker ratio is inversely proportional to the change in flock size. Yeah. Why? Because the remember that the flock zero, the first flock, it's reproducing under the pressure of three predators around. So it will, until the critical point of transition, it will require more soldiers for protection. And here is the same figure for flock zero, the first ring structure. When it reproduces, we see that it is the soldier and worker ratio is proportional to the change in flock size, because we observe that the flock one reproducing in the less threatened environment or surrounding, so it will favor more workers. Yeah, so it's kind of collective adaptation at a collective level. Yeah, we observe it. Yeah, so we have demonstrated an artificial ETI or a kind of ETI in an artificial system. And this process is akin to the transition from solitary to eusocial superorganisms. The whole process is that we have the group formation by a self-organization capacity, which is evolved and selected by natural selection. And we have observed spontaneous group selection. Some groups are better to survive, and there is interdependency within groups. Agents depend on each other to survive and to reproduce. And this makes a collective, cohesive collective. And surviving agents present adaptive plastic division of labor, because we know that the ability to switch between roles is an evolvable ability, because we are evolving neural networks, and neural networks are controlling that ability. And we have observed a spontaneous group reproduction by a process akin to binary fission observed in mitochondria. We see that mitochondrial DNA is a ring structure also, and some comparisons with other frameworks. Unlike kin selection, we don't require genetic homogeneity for groups formation and survival. Unlike multi-level selection or group selection, we don't presuppose the formation or the existence of the group, but the group is spontaneously formed from the bottom up. There are some thoughts after my simulation is that we can ask some questions like what are the critical thresholds for population density and resource availability that facilitate the transition to eusociality. And also we can study how does communication within a group evolve and facilitate the organism of that eusocial superorganism. And I'm more interested in how and can transgeneration and non-genetic inheritance, such as from social learning, can support the establishment and maintenance of eusocial structures. And my whole research before my current postdoc is about the Baldwin effect or the interaction between evolution and lifetime plasticity. In the next step, I will investigate is this Baldwin effect can foster or hinder the process of ETI, especially when the environment changes. So we will require some ability to learn to predict the environmental changes. And here in this current simulations, the predator is just presented as environmental challenges, but it's not evolvable. So the next step is, let's say if we allow the predators to evolve also, and we include the self-organized ability as an evolvable trait like in boys, and can we observe an evolutionary arms race in the transition? This means that the predators should evolve and should evolve the transition into a bigger whole so that they can eat the predators, and then the ring structure should evolve. The ring structure of boys could evolve into a bigger one, which will be the next transition. And also that I'm interested in whether the process of self-organization and maturation in Vitanova can be generalized into other ETIs in nature. And yes, so in this simulation, I'm using a simple neural network to simulate the transition from solitary to eusociality. But in evolution, in classic example of the transition, evolution of multicellularity, the cell doesn't have neural network in it. So how can we extend Vitanova to model multicellularity? And the first thought is that I can use gene regulatory network in place of neural networks. And yes, some other technical stuffs, especially how I need to define more metrics to measure emergent collective behavior, and how can we define the collective genotype from Vitanova. Yes, and measure the difference in change in genotype at the component level of boy, and the change at the genotypic level at the collective level to compare it. Yes, thank you.